Hello, welcome to some video notes. Today we'll be looking at topic 3.5 and we'll be reviewing some of the important techniques that are used in the process of making genetically modified organisms. So we'll be talking again about PCR, something we did cover back in topic 2, but also applies here in topic 3. We'll be looking at gel electrophoresis and how gel electrophoresis helps us to find specific sections of DNA, or how we can use it to create banding patterns of samples in order to compare DNA samples and kind of use them for DNA fingerprinting. And then we'll talk about how cloning is done to a certain extent in the science community to help with help progressing genetic studies.
So you get nothing. You lose. Good day, sir.
So if you remember PCR back from topic 2, you can go back and rewatch some of those notes or look through your own notes to kind of remind yourself what PCR actually is all about. But PCR itself, remember the PCR stands for polymerase chain reaction, and so we're using Taq polymerase, which is the DNA polymerase from the bacterial cells that live at the very, very bottom of the oceans near thermal vents that can survive very high temperatures, have adapted to live at very, very high temperatures near 100 degrees Celsius. And so we use the DNA polymerase, and we can use primers and a sample of DNA, and we can go through these heating cycles so we can make millions and millions of copies of the DNA very quickly, only maybe about 30 or 40 minutes. We can make from one strand of DNA, we could have over 1 billion strands of that same section of DNA. So it's a way to amplify DNA so that we can use it in different genetic studies. So whenever we're dealing with something like DNA and we're trying to create, for example, genetically modified organisms by having the gene be cut with restricted enzymes, and then having the plasmid cut with the same restricted enzyme, then putting those to the plasmid and the gene together and using DNA ligase to make one solid new plasmid. Going through this process of gene transferring, we don't do it once, we don't do it a dozen times, we don't even do it 100 times. We kind of want to do it like 1,000 or so times because there's a very high probability that something goes wrong. So if we're going to work with DNA, we can't really start our process of going through DNA modification or certain genetic studies if we don't have millions of copies of that section of DNA in order to allow us to do the appropriate number of trials in order to ensure that we're actually going to get a result. So that's really what PCR is mostly being used for. It's amplifying a section of DNA so that we have enough copies of that specific section so that we can actually do some reliable genetic research. And to kind of just summarize going through the process of PCR, again, we take the strand of DNA that we want to copy and we're going to heat it so that it will denature. So that is the first phase of our PCR step, denaturation. And so denaturation, so that all the hydrogen bonds between the strands, the double DNA strands, will separate. And then we will allow it to cool down a little bit so that we can get to the next phase, which is annealing. So in the annealing process, DNA primers that have been created in order to match certain sections of this DNA gene that we're trying to copy will stick to the now open free strands of DNA. And then we will then heat up the temperature a little bit more to speed up the last part of it, called elongation. And during elongation at higher temperatures, Taq polymerase is going to attach to the primers, and then it will do what DNA polymerase does. It will read the DNA, it will do base pairing, and so a whole bunch of free nucleotides that are also in solution with all of these materials will be used to make a copy. And so, or make the other side that's missing. So in the end of this, we get a copy of the DNA. So we have one DNA strand becomes 2 DNA strands. And so that's just one cycle. This might take, about a minute. So normally we'll do this, through about 30 cycles. And so from one DNA strand, we can get over a billion DNA strands, like if we if all of these copyings are successful. Now, not every cycle will be successful all of the time. There could be, an instance where we go through the denaturation and the strands of DNA separate, but during the annealing phase, they might come back together again and make hydrogen bonds between them again. That means that the primers can't get in there in order to attach to the DNA. And so that means that particular strands that we just separated, they're not actually going to be copied by DNA polymerase, because the primers couldn't really be there to help DNA polymerase attach to that section. So that's why we're going to be doing this, to try to make as many copies of this DNA as possible, because we're talking about things that are nanoscopic in size, and we can't guarantee that all of these chemical interactions happen exactly the way that would be ideal for us. And so this random percent of error that will happen anytime we go through a procedure like this, we help to reduce that error by doing it many times, which could help to increase the probability that we were successful X number, millions of times. So that means we do have a good number of million or so copies of the DNA that were made properly. So we're going through polymerase chain reaction. So typically uses the copy sections of the whole genome, or we might amplify very small sections of the DNA, depending on what we need it for. So if we're going to need to do Human Genome Project, for example, we're going to all be sequencing the same sections of a genome. Then we're going to have to copy a whole chromosome or whole area of that genome. We're going to need to be copied several times, so that all the different research labs that are helping out with the Human Genome Project, so we know that they're all working with the same section of the DNA when they go through their own sequencing processes. When we are trying to do things like gel electrophoresis, say we want to isolate a very specific gene so that we can use it for gene modification, we're going to have to go through gel electrophoresis to help identify which parts of the DNA where that gene is located, and I'll talk about how that works in just a second. But to do that, we're going to need millions of copies of this DNA, so that we actually have material to work with. If we found DNA at a crime scene somewhere and we wanted to try to use DNA profiling to try to match the DNA sequence of the blood maybe at the crime scene to the DNA that we think maybe came from a certain suspect, again, we need millions of copies of that DNA when we go through that testing to try to make sure that we get some pretty reliable data. And so that's really the whole point of our PCR being used in genetic studies. Now, let's get into more detail about what we mean when we're talking about DNA profiling. And so profiling specifically is where we're going to compare an unknown source of DNA to a potential source of DNA, and we're going to see if they are related to each other based on a banding pattern that is produced. And so this could be used to identify if a criminal might have been at a crime scene, or a suspect, sorry, might have been at a crime scene by collecting some of their DNA and comparing it to the results we get from the crime scene DNA. And this can also be used in paternity tests. So obviously a female will know if she's been pregnant and gave birth, so women often know if they have had a child, but fathers do not always know if they are the father of the child, because of certain things that could occur where there might have been multiple partners that the female was interacting with, and so they don't necessarily know which sperm from which male is the one that actually fertilized the egg, and therefore they are the biological father for that child. And so what we can do is a paternity test, where we look at DNA sequence pattern or a DNA banding pattern from gel electrophoresis for the baby and for the mother and for the fathers or the potential fathers. And essentially, the child should have a mixed banding pattern of both the mother and the father. And so if father C, for example, or potential father C's banding pattern matches most with the banding pattern sections that overlap with the mother, and therefore match up with the banding pattern sections we see with the child, then it's most likely that that individual is the father, because that's how that would occur, is that mixing of their DNA. Half the DNA coming from the egg and half the DNA coming from the sperm. We would expect the child's DNA banding pattern to be a mix of both of their DNA. So before we can really check on all of these results, and we are going to do a couple practices to show what we're talking about, we have to go through the process of gel electrophoresis in order to separate DNA sections or DNA samples by size to create a very specific banding pattern. So going through gel electrophoresis, fragments of DNA will move through an electrified field. And if you remember, DNA is negatively charged. The backbone, the phosphodiester backbone, has a bunch of negative charges on it. So what we can do is, if we put it in an electrical field with a positive end and a negative end, the DNA will move towards the positive end. It will be drawn towards the positive end, right? And so if we have it move through a very thick jelly-like substance called agarose gel, smaller sections of DNA should be able to move through this gel faster than larger sections of the DNA. So what we could do is we could separate all of the DNA in that sample by size. And so the smallest pieces that we've cut up using different enzymes would be the furthest down the gel, or would be closest to the positive end. And the largest ones might not move very much at all over a very specific amount of time. So when this occurs, and then we kind of color them with a specific dye so that we can see the patterns, the banding patterns very clearly, it creates sections of large and small blocks of DNA, and that's what we call a banding pattern. And so someone who has the same DNA as DNA that we found at a crime scene, if we use the same restriction enzymes and we cut it in the same places and we run it on the same gel for the same amount of time, they should have identical banding patterns, because the DNA would be separating into large and small sections exactly the same, because all of the sequences and the size of the DNA samples would all be the same from that individual. So we're going to go through this examples in a little bit, but basically run through the steps, and then we'll look at it in a little bit more detail on a diagram. So the first thing we have to do, if we're going to do gel electrophoresis and profiling, is we're going to have to do PCR, right? Because we're probably not going to have many copies of the DNA, and we need millions of copies of the DNA in order to make sure we get solid results. Plus, the more copies of the DNA that we have, the darker the bands are going to appear in the gel, which makes it easier for comparison. So we're going to do PCR to amplify the DNA. Then what we're going to do is we're going to cut the DNA samples with the exact same restriction enzymes. And if you remember how restriction enzymes work, they will find specific sections in the DNA, specific nucleotide orders, right, sequence order, and it will break the phosphodiester bonds at that point, right? And sometimes it will make flat ends, sometimes it makes sticky ends. But the thing is that these restriction enzymes are extremely consistent. They will only cut the DNA at that specific sequence. So if I have my sample from a crime scene, and I have my sample from my suspects, and I use the same exact restriction enzymes on all those samples, if the DNA at the crime scene is the same as the DNA from suspect B, then they should be cut in the same places. This means that the sections of DNA, the large and small little pieces that form from cutting the DNA with these restriction enzymes, should be the same. So then what we can do is we can add a little bit of a fluorescent marker, which will bind to the DNA fragments so that it makes it a little bit easier to see them, inside the gel. And then what we're going to do is we're going to load the DNA into one side of an agarose gel, and we're going to run electric current through it. And so it's going to move from the negative end to the positive end inside this electric current in order to because it will be drawn towards the positive end. And as it pushes through the gel, larger or heavier sections of the DNA that were produced by cutting it with the restriction enzymes are not going to move very far, and the smaller ones are going to move very, very far. They're going to move the furthest. And so by separating the smallest sections of the DNA produced versus the largest sections of the DNA produced, we get that banding pattern. And so then all we have to do is compare the results of the banding patterns that are produced, and then that will tell us whether or not we're looking at, potentially a father for paternity test, or a suspect might have been at a crime scene. So let's look at that a little bit more clearly with maybe this diagram might help explain it a little bit better. So this idea, again, we have to take our DNA sample, then we're going to have to amplify it using PCR to make millions of copies of the same section of DNA. And then what we're going to do is then mix those sections, all those copies that we've created, with these restriction enzymes, which will cut it in very specific locations in the DNA. And because sequences are all different from each other, the number of short sections versus the number of long sections and the size of those short and long sections would be different from one person to another. And so this creates a very specific cutting pattern or banding pattern because of cutting the DNA in very specific places. So then what we do is we add the samples to a gel electrophoresis well here. And so inside this area here, this kind of grayish area, that is an agarose gel, which is a pretty thick gelatin substance. It's not very easy for things to move through it because it's very dense. And then we are inside this container where everything is placed inside of an electrolyte solution, so electricity very easily passes through it. And we have a negative end where the samples are going to be loaded, and then we have the positive end. And so when we run an electrical current through it, the current will move from the negative end towards the positive end. And when it moves through this solution, it will also move through the gel, it will move through the DNA sections inside the gel. And so because of the negatively charged backbone of the DNA sections, of the DNA strands, they will be drawn towards the positive end of the container, and so they will move through the gel at a different rate. And so you end up with a situation something like this. And so here we've got different suspects. You could think of it as a paternity test situation. And so here we're comparing individuals 1, 2, and 3, and then we also have a size marker as well. And so a marker is also known as a ladder because it kind of makes like a ladder shape, inside the gel. Has very specific sized banding patterns. So for example, this one all the way down here, this might be like, oh, we're only dealing with something that's 100 base pairs long. Where this one up here, being very, very large, maybe that's like 10,000 base pairs long. Okay? And so if we're looking for a specific gene that we want to isolate, for example, maybe if we're trying to use this technique to find the human insulin gene, and we know that the human insulin gene is maybe about, 5,000 base pairs, right? And this is the place where the 5,000 base pairs might be, right? Maybe that's what that banding pattern number is. Well, then that tells us like, oh, these ones right here, those are where we might find our human insulin gene. And so we would cut these specific sections out. And then what we can do is we can actually dissolve the gel really easily, and then we can get the DNA back out of it. And so therefore I have isolated the human genome the human gene of insulin that we are specifically looking for, and then we can use it to, go through and add them to plasmids and go through the whole transformation of the bacteria using a recombinant plasmid and make a whole bunch of human insulin, right? If this is for a paternity test or for a crime scene, then we would just be looking at the overall banding pattern. So for example, let's say 1 was the DNA found at a crime scene, and 2 and 3 are both our suspects. Well, it looks like the banding pattern for 1, 2, and 3 are not very similar to each other at all, so probably none of them, 2 and 3, were not the people that were the source of the DNA that we found at the crime scene. Or this could be something like a paternity test. So let's say here, individual 3, let's say that person was the baby, right? And individual 1 and individual 2 are the father and the mother. And we see that there are some similarities. We get this band here, right? We've got this band here, right? But, okay, so if this is missing, this could be coming from there, right? But then we have this other band here at the bottom that doesn't seem to be coming from either individual, right? So if this 2 is the mother, right, then that means that this band here would have to come from a father individual. And so here we can see that there is no band present in this part of the gel for that potential father. So it's possible that this person is not the father because the banding pattern isn't quite matching up. Now, in this example that we see here, this is just to kind of give you a visual of how smaller things move further down the gel versus larger things. If we were to do a real paternity test, there's way more bands that are produced in an actual paternity test. So it's there's a certain probability that goes along with this of how many of the bands are the same versus how many bands are not the same. And they could present a certain probability of like, oh, there's a 98% chance this person is the father, or there's a 38% chance this person is the father. Based on, again, we're dealing with nanoscopic materials. There are, certain random errors that could occur. So it's very difficult for us to say 100% sure someone is the father, but we can be very, very sure that someone is the father based on the banding pattern. So here again, the reason why we can see them in the gel, the reason why they look like they kind of glow a little bit is because they are also tagged with a fluorescent compound, so that when we put them under, for example, UV light or some type of fluorescent light, the tag picks up the energy from that light source and causes it to glow. So it makes it a lot easier to see the sections of DNA because all we need to do is put it on a nice light source and when it will glow a little bit, and so we can see the banding patterns a lot clearer. And then, of course, we have a marker here to help use to find specific DNA fragments based on their size. And we might be looking at the number of shared banding patterns, again, if we're looking at something that is a paternity test or looking at evidence at a crime scene and trying to match that to a suspect. Okay, so this is the general steps of how gel electrophoresis works. And now we're going to get into a little bit of some practice thinking about how it applies to paternity tests. So DNA profiling is often used in paternity tests and forensic investigations. DNA is often left at crime scenes. It's normally criminals might be getting a little bit smarter about things because of like crime shows like CSI, but typically people leave a lot of evidence that contain DNA around at a crime scene. They might leave skin cells, they might have cut themselves at some point, so there will be blood present. They might have lost some hair follicles, and there could be some hair cells at the bottom of the strand of hair, right? And if it's something that might be a sexual assault, of course there could be semen that's left behind, right? And if I give you an example, the 1986 forensics DNA analyze was first used. It was originally known as DNA fingerprinting, and it's this type of DNA. Now we call it more like DNA profiling, not to confuse it with fingerprinting, but more DNA profiling or DNA testing to try to narrow down what's the possibility of the evidence that we found at a crime scene coming from specific suspects, right? And so it is easier to exclude a suspect than convict someone based on DNA matches, because the FBI estimates that about 1/3 of initial rape suspects are excluded because DNA samples fail to match. So when we use something like DNA profiling, it actually helps the police officers very quickly to try to determine who shouldn't really be a suspect rather than who should be a suspect. So we can very easily see when banding patterns don't match, but it's very rare that we have banding patterns that 100% match, right? So we then have to look at, well, what probability is it that this individual has their DNA matching, their DNA matches what we found at the crime scene, right? So we can't just maybe base an entire case on DNA profiling, but it does help police officers very quickly start to eliminate suspects so that they can focus on other individuals that did not, pass the DNA profiling scan. And then it can also be used as supportive evidence in the court of law to help add more probability that the individual that the suspect that we have, their DNA does match the DNA that was found at the crime scene. Okay? So, and forensic investigators take a lot of precautions to make sure that there are no mistakes and that there are no human error either in the collecting of the DNA or the processing of the DNA through PCR or the processing of the DNA through gel electrophoresis. And so there is a certain amount of human error that can occur. That's why it's quite difficult to say the results are 100% accurate. Because again, we're dealing with something it's nanoscopic. It could be very easily that someone who's working with the DNA samples, their DNA accidentally gets into the samples that they're working with because they lost some skin cells, or, you know, they sneezed and so some of their cheek cells kind of came out through the process of sneezing, and so it ended up in some of their samples. Now, when we look at paternity tests, we can take samples from the mother, we take samples from the child, and we take samples from all of the fathers who are the potential fathers that we think. And then we look at inheritance probabilities, right? So we look at dominant recessive traits inside of their genes, and also just dominant recessive traits they themselves possess. So for example, if we've got a mother with brown eyes, and we've got a baby with blue eyes, and we've got a father with brown eyes, but both of his parents have brown eyes, then there's a really high probability that that person might not be the father, because it's unlikely that that person is heterozygous for brown eyes. So that means the child having blue eyes would not really be possible. But that can't be the only thing that we base our paternity test on, but it's something that has to be considered. And then we'd also look at just the results from the paternity test to determine if this person is the biological father based on our banding patterns. So now we can go through a couple practice problems to kind of show you what this would look like. Okay, so DNA profiling here, looking at this instance where we've got a case where there was a crime scene and there was a hair follicle was left behind at the crime scene. And so we need to determine who owns that hair cell, right? Where did that DNA come from? So in column A, we have the evidence from the crime scene. In column B, we have the homeowner's own DNA. And then C and D are the two suspects. And so looking at the banding patterns, this is a little bit more idealistic, but looking at the banding patterns, who matches with A, or what matches with A, are evidence at our crime scene. All right, figure it out. Yeah, so it's C. We see that the banding patterns are matched here and here. There are no other bands matching with B or D in our evidence A, so this seems very clear that a suspect C must have been the person who left that hair at the crime scene. Now, that doesn't guarantee that suspect C is the one who committed the crime. There might be a logical reason for why suspect C should have been there. Let's say that this guy was a delivery man, he was a mailman, right? Depending on where this hair was found at the crime scene, maybe it makes sense that his hair would fall out in that area if he was just doing his regular job. So this might give evidence to help support the idea that that person was there, but it doesn't necessarily prove that the person committed the crime. So the other evidence has to be collected in order to really convict someone under these circumstances. Okay. So here's another example. So in this case, there was a lot of blood left at the crime scene, and so we're trying to identify who is the owner of that blood. So we've got A, which is the victim, person who is from the crime scene, B, which is an unknown blood sample at the scene. So we're pretty sure that blood found from person A, who is the victim, and then we have B, which is our unknown person, and then C and D, we have our suspects, right? Suspect 1 and suspect 2. And so then looking at the banding patterns, where do we see the person matching up, or who do we think matches up with the blood at the crime scene? Right? Figured out that it's C, right? We see a lot of bands matching up here, right? And so we know that there is an unknown blood sample, because here we've got A and B are matching a little bit, but we've got these two different samples here, right? There's two different banding patterns there. And that shows us that that blood isn't entirely the same as the victim. So there must have been another individual that might have been at the crime scene, might have been the blood of the person committing the crime, right? So again, it doesn't entirely say that the person did commit the crime, but it does say that person must have been there for their blood to be present at the crime scene. And so we need to investigate, why would that person's blood be there? What could be the possible reason behind it? So in this case, DNA evidence that was being used in a wrongful conviction case. So is the prisoner really guilty? So we've got our trace evidence in A, the homeowner is B, and the person who is in prison for this crime is C. And so do we feel that the person was rightfully convicted under these conditions? Probably not, right? So if you think about it, There are some overlapping sections. Here, A and C are overlapped, right? Here, A and C overlap here. But there are numerous banding points, right? Actually, more banding points than not, that don't match with the evidence found at the crime scene. So because there isn't really a very strong match, right, or majority of them do not match, there's probably a much higher probability that that person was not at the crime scene, and then that someone maybe made a mistake in the process of doing this investigation, and so this person probably needs a new trial with more to take in more evidence to consider whether or not the probability that if they weren't there, maybe what do they have a good alibi that the DNA can then help support to show that they weren't at the crime scene. So there are various cases that are appearing now in the last couple of decades of people getting, and when we say exonerated or getting released from prison because DNA sampling has shown that, oh, they weren't in contact with this person, or that wasn't their blood, or they weren't lying, or their alibi was very solid, and the DNA shows that they weren't actually at that crime scene. Okay, another one. In this case, looking at parentage for a child. And so we have A is the mother, B is the child, C is possible father 1, and D is possible father 2. Now, when we're thinking about paternity tests, we're looking at B, and we're trying to see, is B a relatively even mix of both A and another person? And so you want to see for banding patterns where the child would have got their DNA from their mother. So here's the banding pattern, banding section where their DNA came from their mother, right? And so do we think someone might be this child's father, or who could be this child's father? And so then we look at the other bands that are present that don't match up with the mother, but do they match up with another person? And so here these match up with C. This one also matches up with C. So it looks like B is a even mix of both the mother DNA from A and the father, who seems to be person C. And so this is very similar to how a paternity test would look. We'd see how many of these bands that don't match up with the mother match up with the other potential fathers to help indicate, oh, where did the other half of this person's DNA come from? Okay, and then one more practice question. So you can take a moment, you can pause this, all right, see if you can answer these questions. Okay. All right, so question 1, identify the smallest DNA fragment. And so remember, if we're going from our starting point to our ending point, here's our origin, right? This is where the DNA started. That means that band 5 would all have the smallest DNA fragments because the smallest DNA fragments will travel the furthest through our gel, and so they should end up furthest away from the origin point. Next, state the number of bands that would appear in the standard lane. So how many standard bands would we want to have present if we're going to be doing this paternity test with all the different points that we've cut it? So when you think about it, how many bands do we have? We've got 1, 2, 3, 4, right? So we'd want at least 4 bands in our standard line or our marker line, right? So that we can compare band size and give us an idea of how big the DNA must be in those bands that have appeared. And then identify the child most likely to come from the mother's previous marriage. So if we've got the mother here, right? And we've got child 1, child 2, child 3, child 4, and we also have their father, right? And so one of these children may not have come from a combination of the mother and the father, but from the mother and another man that was from a previous marriage. And so we have to think of, where is all the DNA present from both the mother and the father? So going child by child, if we start with child number 1, right? So child number 1 seems to match up here and here with the father, here and here with the mother. So that seems to be okay. Looking at child number 2, child number 2 matches up here with the mother and here with the mother, but there are no bands at all matching up with any of the father's DNA. So that looks like child 2 is the one that must have come from a previous marriage, because we don't see any banding patterns similar to the father. So we only see it matching mother's DNA. Okay, so hopefully these practice questions are pretty easy and help give you an idea of how this information can be used on our test. Okay, so the last part of these notes, we're going to go through the ideas of what clones are, and then how we can purposely create clones in order to help with genetic studies, and also, helping to make genetically modified organisms. And so a clone is a group of genetically identical organisms, and a group of cells derived from a single parent cell. So you could think of, for example, if I take a single E. coli cell and I grow that E. coli cell out to millions and millions of copies, all of those copies of all those millions of bacterial cells are all genetically the same. All right, they're all identical to each other because they all came from the same original cell. So they are all clones of each other. All right, you could think of your body cells are all kind of clones of each other when they go through the process of mitosis. When we want to repair your skin cells when you get cut, the process of mitosis makes a genetically identical daughter cell. So those would be considered clones, right? So there are different ways that these can occur naturally in the world. So first off, we can have monozygotic twins, which means that monozygotic twins, they were a single zygote that split into two during the process of it moving through the fallopian tubes and before adhering to the uterus. So rather than it being a single ball of cells that grows into a fetus, you get two separate balls of cells that eventually grow into two different fetuses, and they will be genetically identical to each other because they are from the same sperm-egg combination, so they should be all sharing the same DNA, right? However, they don't necessarily continue to, as they get older, always look exactly the same. Identical twins or even triplets, they do look identical very much when they are born, but as they get older, epigenetics comes in and starts to modify which parts of their DNA are overexpressed or underexpressed through things like methylation and acetylation. So we all have our own epigenetic occurrences depending on our interactions with our environmental conditions. So there can be a certain amount of time that identical twins will look identical, but eventually they will start to, on a genetic level, start to express themselves a little bit different. So they will start to look a little bit different from each other as they get older. There also is cloning is used a lot, of course, in repairing our tissue. For example, starfish, if they get damaged, if they lose, a part of one of their arms, they can regenerate that whole arm in order to, replace it through this natural cloning of a section of their body. And of course, through asexual reproduction, through binary fission with bacteria, all of that is cloning as well. So all the bacterial cells in a population, if they all came from the same bacterial source through binary fission, they're all going to be clones. Now, when we turn to things about agriculture, there are some naturally occurring clones, like things like runners. So runners might appear to look like they are individual plants because under the soil is where they're all connected to each other. But above the soil, they all look like, oh, that's a plant, and that's, this is one, and this is one, and this is one, and this is one. But under the soil, they're actually all connected to each other by the same root system. And they're actually extensions of one organism, and each one of those sections are all being reproduced asexually. So they are not new plants. I mean, they are new plants, but they are not genetically new. They are all identical. They're all clones of each other. And so this allows the plant to propagate very, very quickly. It can make many, many copies of itself, many extensions of itself, and that can help with its success. Being a faster growing plant might be the whole reason why you're more successful in an environment versus another one. Another example of this would be things like tubers. So things that are like, for example, potatoes, right, or turnips underground. They have these little sections on them, which we often call the eye of the potato, right? They look little brown spots like that. So that's where all of the leafy section of the plant is going to grow. And the potato itself, that's the really starchy storage center for all the energy that's been produced by photosynthesis so that the plant can survive, going through winter and things like that. But from these little spots, there will be multiple plants growing, but all of those plants are genetically identical to each other. They are all clones of each other. All right. So during the winter, right, we'll lose all those plants, and the tuber will just stay underground, waiting for the temperatures to warm up and for the days to get longer. And then you'll get multiple potato plants sprouting up under the soil, but actually, if you trace them all back if you dig it all up and you look, they're all connected to the exact same potato section, and all of those individual eyes, or those individual branching points from the potato, they're all identical to each other, so they are all clones as well. So with monozygotic twins, so it's possible that these cells could occur through, or monozygotic twins could be created artificially if we wanted to, and this actually does happen in some parts of the world. For example, there was a time period, they've recently changed it, but there was a time period where China had a one-child policy. But of course, that one-child policy didn't apply to twins. So there were a lot of people, they really wanted to have, 2 kids, and they wanted to have a brother or a sister for their child. So they would make sure they would pay extra money to go do IVF treatment. And during the process of the IVF treatment, they would have clones be created. They would have twins be created through a situation like this, where you have the balls of cells, you have the little blastoids here, and then they would use a very, very tiny needle, and they would steal some of the cells from inside of that original zygote, and they would then implant them in another emptied ball of cells, and then we could implant both of them into the uterus. And so both embryo A and embryo B are going to be genetically identical to each other, and so the woman ends up having twins instead of having a single baby because of this IVF treatment. So if we wanted to clone a human being, that's kind of some way that we could do that. We could, separate the cells of the blastocyst so that we get, if we make a monozygotic twin, rather than just having it occur naturally, we force it to happen. We could also use the process of the natural occurrence of clones in order to help us with our scientific investigations. So one of the things that we could do is plants that are runners, for example, that root very, very easily. If we cut them at just the right point along the stem, they can then regrow the section that's been missing in order to make themselves new roots. And if you do this repeatedly over a couple of samples, you can then get a whole tray full of various plants that are actually all genetically identical to each other. And so this would actually be a really good way to control variations in the DNA when doing some type of study on plants. So for example, say we wanted to investigate the effects of different fertilizer, or we wanted to investigate the strength of the sunlight, or the concentration of CO2, or other dissolved nutrients, or effectiveness of how long the leaves are in terms of photosynthetic rates, right? We could look at the same species of plant, but all of those individuals in our sample might be genetically different from each other, and so that becomes a variable that we can't control. We can't necessarily guarantee our results are really because of the variable that we changed, and not because of some genetic differences between the one population that got more light versus the other population that got less light. So if we were to use clone individuals like runners, for example, and we were to cut them in just the right way and then replant them, then all of those individuals would be genetically identical to each other. And therefore, when we put them through these different stimuli, we can remove genetic diversity as a factor from our results, therefore being a little bit more sure that it is the variable that caused the results, not some differences in the genes. So we talked about cloning that occurs naturally in animals, cloning that occurs naturally in plants. We talked a little bit about how cloning could be done artificially in an IVF treatment to make a monozygotic twin. And of course, we could also use naturally occurring plant clones, or help to cut those types of plants in just the right way, so that we make a whole field of plants that are the same, that are genetically similar for studies. And so the last thing we're going to talk about is how could we use some scientific techniques to actually legitimately clone an animal. So how can we get a very specific trait to be continuing on from one process to another? And so we're going to get into how we could clone differentiated cells. And so let's say we have an organism that has very specific desired traits, and we want those traits to continue. The problem is that if we allow that organism to reproduce with a different organism, only half of their DNA is going to be contributed to their children. And so some of the traits that we really, really want, they might start to disappear, they might fade, they might, get mixed up with the DNA of the other parent. And so we're not really keeping this kind of pure genetic trait line or gene line for those traits that we really want to exist in our population. And so if we really want the specific trait, what we could possibly do is use somatic cells of an organism, and we could induce them to be kind of pluripotent and help us to create an organism that would be genetically identical to the original organism by taking the DNA from body cells and then helping them to become pluripotent and by inserting them into cells that are going to go through the process of pluripotency. All right, so maybe induce pluripotency in these cells. So one of the ways we could do this, or the way we do this, if we go back to 1958, so John Grudon basically came up with this method of transplanting a nucleus into an unnucleated egg cell. And so the chemical signals inside of the egg, right, helps promote this rapid mitosis to create and to use the DNA that's present in the nucleus to create a full organism. So the thing is that when the egg becomes fertilized, right, and so it has a full diploid nucleus, then it is triggered to go through this process of, okay, let's make many, many, many copies of this cell through mitosis, and let's grow this whole organism. Let's read all the DNA, let's differentiate to all the different types of tissues, and let's grow whatever it is that that we've been designed to grow. What is the DNA that we need to grow, right? So then this idea is, well, wait a minute, rather than having a sperm mixed with an egg, right, and having a new diploid cell created by the mixing of those two cells, what if I removed the haploid nucleus from the egg, right, from the egg, and then I inserted a diploid nucleus from a different organism, right, same species but a different member of that species, would the egg therefore use the diploid nucleus that I just gave it to then go through the process of differentiation and growth to create an individual that is genetically identical to the sample that I just took from? So this idea of, rather than using, for example, a mother's egg cell and a father's sperm cell to make a baby, what if we took the diploid cell from the father and put it into the female's egg? After we've removed the nucleus, would we just then make a genetic clone of the father? And that's kind of actually how it works. And there's this process called somatic-cell nuclear transfer. And so if you've ever heard of Dolly the sheep, right? Dolly sheep is the one that was cloned using, it was their first animal clone using this technique. And so essentially, it's not that complicated of a process. So what we have to do is we have to have a diploid cell that we want to, that comes from an organism that we want to clone. So for example, if this was Dolly the sheep, they took breast tissue from Dolly the sheep, or from the mother, I guess, of Dolly, or the original, or the uncloned version of the sheep. And so they took the breast tissue from that sheep, and then they isolated just the diploid nucleus by doing very tricky procedure with very, very fine needles. They inserted the needle into the they inserted the needle into the breast tissue cells, and they just kind of sucked just the nucleus of the cell away, and then they kept everything else inside of the cell. Then they took a donor egg from another sheep, and then they removed the nucleus. They enucleated it, right? They took the nucleus away from that donor egg. And then what they did is then they inserted the diploid nucleus that they took from the original sheep into this donor egg, right? And so that means that we now have an egg that is fertilized in some way with the new diploid nucleus that came from the original sample. And so then this now fertilized egg, this now diploid sexual reproductive cell, is now a zygote, right? And so it can go through the process of growth and development, mitosis, differentiation, and it can grow a new organism. In this case, it grows a new sheep. So then the implanted new zygote is the zygote is then going to be implanted into a surrogate. all right, and then it's eventually born. And if we do genetic testing, the newborn baby is going to be genetically identical to the organism that we took that original nucleus, diploid nucleus, from. And so when we think of Dolly the sheep, we created a clone of a sheep using this whole process. So we're taking the nucleus from the mammary cells of one sheep and then putting them into the egg of another sheep after we've taken the original nucleus out. The new diploid zygote, right, that has been created was then implanted into another sheep, and then after it was born, we had an actual clone of our original sheep, right? And so that is all how this process is possible. So using this technique, we can create genetically identical animals, or clone animals, or clone whole organisms, again, for this whole process of helping us along with genetic studies, leaving or removing genetic features out of the equation. We could also do this for helping with selective breeding, if we're trying to create better crops, if we're trying to create better herds of animals, and we have specific traits that we don't want to get lost through the process of breeding, we could use this technique to clone animals in order to help keep the genetic lines, unmixed for future generations. And of course, there is now more of a modern way that this is getting used, where some people are cloning their pets. They're cloning their cats and their dogs so that, after they've died, they can then have a new cat or a new dog that is essentially the exact same cat or dog that they had in puppy or kitty form, because it would be very young compared to the one that just died. But genetically, they are exactly the same. Now, there's a downside to this, however, because even though this technique is actually not super difficult to do, it has a pretty high success rate when you get the right materials, clones do not live as long as their original organism that they would have been, their information would have been taken from. We're not entirely sure, but it probably has to do with the quality of the DNA inside of that original nucleus, the diploid nucleus taken away. But clones typically live a half or sometimes even less the lifespan of the original species, the original organism that has been cloned. So we haven't really been able to combat the effects of aging, even though we have figured out a way to clone an organism. So again, going through this process, again, outlining these steps, removing the diploid cell, enucleating an egg cell, reinserting it, allowing it to grow in vivo. But then in this step, rather than inserting this embryo, this zygote, back into, inserting the embryo, because we do get the embryo phase, inserting it back into a surrogate, what we could do then is to break open the embryo and to collect the stem cells from the embryo. So this also means that we've created a source for stem cells rather than having to destroy a potential embryo that would have continued to live on and create a whole new human being, for example. What we could do is use a regular diploid cell from a regular person. We could use some donated eggs that somebody had no intention of using because they're just donated eggs from another female. And then going through this process, we could start to create human stem cells that come from a source that maybe is doesn't really cross any ethical lines, because we're using eggs that were never going to be used anyways, and we're not even using a sperm. We're using a diploid cell, which came a nucleus from a diploid cell, which came from the blood or the skin of some other human being that donated some material to science. And so it is a potential way for us to create stem cells that are a lot less ethical and are a lot easier for us to maybe control what exact genetic information is inside those human stem cells, so that we can help us with our genetic our testing when working with stem cells. Okay, so uses for this, obviously, creating stem cells for transplants for patients who need skin grafts or are dealing with leukemia. So we could create stem cells specific to you, right? We could try to clone your bone marrow stem cells, for example, or clone stem cells that would match your genetic information. So then when we insert those stem cells into your body, they're not going to be rejected because they are built from your DNA. So that means your immune system isn't going to fight them off. We could use this for creating replacement tissue that, again, is genetically identical to that tissue. So if we need to grow somebody a new pancreas or build them new nerve cells, it would be built on their own DNA. So therefore, it should fix and work with their body perfectly fine because they're all using the same DNA. So this obviously reduces the risk of cells or tissues or whole organs being rejected by the body's immune system because genetically they are the same. Okay, so that has been this series of notes on 3.5, and hopefully if you have any questions, you can ask me.